Hey guys, welcome back to my channel. Today I want to talk about cytoskeleton. So for MCAT, there are three types of cytoskeletons you want to know. The first one being microtubules. So this is going to be the topic of today's video. And then in a couple of days, I'll upload another video that talks more about microfilaments and intermediate filament. All right, so I want to give you a general sense how cytoskeleton looks like in a cell. As you can see, the green ones are microtubules, which we're going to talk about their specific structure and their specific functions. And then the blue one is intermediate filaments. They're more spread throughout the cell. And then lastly, the microfilaments, or the purple ones, they're located more towards the cell membrane. All right. So the three types of cytoskeletons, they do share common function of supporting the structure of the cell, but they also have their unique structure and functions. So today, let's dive into microtubules. The structure of microtubules is that they have these smallest building units called tubulin. And then this is the alpha type tubulin. This is the beta type. When alpha is attached to beta, as you can see here, they form a heterodimer structure. And as you extend this heterodimer structure, you actually form something called protofilament, which is confusing because here, this word has filament in this word. But actually, it's just a subunit of microtubule. It has nothing to do with microfilaments or intermediate filament, right? All right, as you can see here, another picture that I found. First is the alpha tubulin connects with the beta tubulin. They form this heterodimer. And then as the heterodimers continues, we have this protofilament. And lastly, 12 protofilaments make up the microtubule, right? That's the structure of microtubule. Cool. All right, now let's talk about the functions, unique functions of microtubules that other cytoskeletons don't share, okay? So first is microtubules are heavily involved in cell division, all right? We're gonna dive into the details in just a bit. And then secondly, microtubules are very important substituents of eukaryotic cilia and flagella. And then lastly, microtubules are very important for axonal transport, okay? So let's look at cell division. And then before cell division, let's look at DNA replication. As you can see here, I have this chromosome that hasn't been replicated yet, right? So it's just one double strand over there. And then after replication that happens in interphase, I have two sister chromatids, right? These two sister chromatids are exact copies of each other, okay? And then in the middle of this chromosome, I have a region that is called centromere. As you can see, I have two proteins here at a centromere region, and then they're called kinetochores, okay? We're gonna mention kinetochores in just a bit. Okay, so now let's go back to interphase. So during interphase, not only did chromosome replicate, but also some other stuff in the cell, they also replicate themselves, okay? Specifically, I mean some stuff at this centrosome area, okay? Centrosome is a region, it is an area in a cell that's near to nucleus. And inside, at the centrosome area, what do we have? We have two very important organelles. They're called centrioles, okay? And then we have a lot of microtubules that's attached to the centrioles that we're gonna see what they do in just a bit, okay? So these are, the black highlighted are centrioles, okay? And then these pink microtubules, they are called astromicrotubules, okay? Why are they called astromicrotubules? Because all of those tubules and centriole here, they're called aster. They look like a star. So all the stuff in the centrosome are called aster. And then those microtubules are called astromicrotubules. All right? Okay, so during cell DNA replication, not only does DNA replicate, so does this aster. Okay, so let's make a copy of this. Okay. Let me flip it. Okay, Now we made a copy of it, okay? And then I'm also gonna draw some random chromosome that went through replication, okay? Now let's move on to the metaphase of mitosis, okay? During metaphase, we know those sister chromatids, they would line up in the middle of the cell, okay? And then now what happens is the microtubules that's attached to the centriole, they are gonna start to go out. And then what they do is they attach to the chromosomes on the kinetochore attachments. So kinetochores are the attachments of the chromosome and the microtubules. And then these orange microtubules, they have a name called spindle fibers, okay? So they're really the very important microtubules that does the job of separating the sister chromatids during anaphase, okay? Okay, so this is one of the most tested function of microtubule, that is being the spindle fiber that separates sister chromatids, okay? And then this whole setup, including the centriole, the astral microtubule, and then also the spindle fibers, it's called spindle apparatus or mitotic apparatus, okay? All right, so let's just go over again what's in the spindle apparatus. We have this pink astral microtubules, okay? And then we also have the centrioles that has microtubules that's attached to it. And then during metaphase, the centrioles can send out those microtubules to the attachment point, which is the kinetochores of the chromosome, so that the spindle fibers, the orange ones, can contract themselves. They can shorten themselves to separate the sister chromatids, okay? So that's the spindle apparatus, okay? All right, and then the second function of microtubule is being the important substituents of eukaryotic cilia in the flagella, because in the prokaryote organisms, they really have different structure of their cilia and flagella. But let me show you the structure of eukaryotic cilia and flagella. As you can see, this C-section of the basal body, we have 9 outer doublet microtubules, right? And then we also have this central microtubules. So this is really the 9+2 arrangement, okay? Being you have 9 things outside, and then each one of those 9 things has 2 microtubules. All right. And then lastly, the function of a microtubule is axonal transport, okay? So let me show you a picture of a neuron. So as you can see, this green neuron, on the left, it's a soma, right? It's the cell body of the neuron. On the right side is the dendrites. So axon transport really means transporting stuff either from the soma to the dendrite, which is the forward transporting, going through the axon, which is in the middle, right? Or backward transportation from the dendrites to the cell body or the soma, okay? So what microtubule does here is it's really just like the bridge linking the soma and the dendrites. As you can see, we have those gray stuff here in the picture. They're the microtubules, right? And then there are 2 proteins, kinesin and dynein. They're transporting a whole bunch of stuff like the neurotransmitter vesicles, mitochondria, a lot of other proteins that they're transporting. But the point is they cannot do this without the microtubules, right? Microtubules are the linking bridges between the 2 ends of the axon. Of the neuron, I mean. All right, so I hope that's helpful. That's all about microtubules I want to talk about today. I hope that's helpful.